So I'm going to talk to you about glycoinformatics and try to describe the landscape of what we're doing. And to that end, I will start with a comment as a source of inspiration, especially in my group. We have been very influenced by the organization in Swissprot. And in Swissprot, as you probably all know, I mean, Uniprot and the annotated part of Uniprot, a protein is actually described through a number of sections. Whether, I mean, you can have information on the 3D structure, you can have information on variants, on post-translational modification, and this is surrounded in highly populated environment of databases, and also databases with toolboxes, prediction tools. And with prediction tools, you have usually some scores that allow you to trust or not trust a prediction. So this is very rich, and for the past 30 years, certainly has evolved in making the information in Swissprot very rich. So we wish we had a similar situation with glycans, where we can describe the structure, the binding, its involvement in disease, or the expression, and so on. But we have a lot of gray area. So we do have databases for sure, and this is what this course is going to be about, introducing you to some of the resources that we have. We do have some tools, but we miss a lot of the expression data measured in a way that is going to be informative. We need a lot of other prediction tools that are in the making or not even made, etc., etc. So this is basically where we can start. And the explanation for these missing bits that we have is that the accumulation of data, experimental data regarding glycosylation, has been achieved with different means. And so you have the protein scientists who are interested in the protein, but they're not really interested in the sugar. So they do site mapping, irrespective of which structure is sitting on the protein. You have a lot of experimental data that are used to solve the structures, NMR, HPLC, mass spectrometry, etc., etc. And so this is an expertise in itself to actually solve these structures. And the part on affinity, so which are the molecules and the proteins that recognize the sugars, that bind the sugars, is another side of experiments as well. So as a result, we end up having data put into silos, but not really talking to one another. And so this is what glycoinformatics is trying to solve. And because there's so much to do, we decided very early on to try and join forces with the different initiatives that have started in 3 places, basically, in terms of trying to integrate data. Although I should mention that there was an early initiative in Germany by the group of Willy von der Liet, who deceased, and then his group was actually disappeared, although one man is trying to hold it together, Thomas Lütteker, and I'm not really including him here, but he should be as part of the landscape as anybody else. And we, as these 3 recent initiatives, decided to join forces. And one of the reasons why we can do that is that we speak the same language. I'm going to repeat somehow what Serge said before with the description of the molecules, and the fact that we have this notation that allows us to describe in cartoons, which are much more easy much easier for non-carbohydrate chemists to understand. And we have here, I put this as a lot of the experiment, unfortunately, only give us the composition in monosaccharides. So this is the notation for composition, where you can't actually say it's an exose, but you can't say which color it is. And this is the notation. So we try with this small community of glycoinformaticians to put our resource together and also to standardize, to promote standards so that at least we can communicate across our different resources. And Kyoko, for instance, has put together some converting. I mean, we also have converters. We keep on having to convert formats because no one is using the same, or no one. We are trying, but a lot of people are in isolation, don't know which format to use. So we need these converters. And in that paper, Kyoko summarized very well the fact that we have mainly the IUPAC format, some Japanese format here, linear code, the GlycoCT that was launched by the German group. And so all of them are either text, but also tables. They account, so they have different properties. They account for repeating units or cyclic units or not. They accommodate rare monosaccharides or not. And actually, I smiled when Serge said that the GlycoCT was human readable. It's not ticked in Kyoko's table, and I think it's not very human readable, unless you know what you're talking about. Anyway, so we have these different options because carbohydrate chemists have put a lot of effort into this, but the protein scientists have put other efforts, metabolic specialists have put other effort, and so on and so forth. So this is why we end up with many different formats and different ways, but we try to put them together so that it makes sense. So Kyoko will present her Glycosmos portal after this talk. Early this afternoon, so at 1 p.m., the Glygen group is going to present their initiative. And I am going to briefly talk about our initiative on XPC. So for those who are, of course, since you are following a SIP training, you probably know that SIB is doing a lot of bioinformatics for quite a while. We celebrated the 25th anniversary a few years ago. And XPC has been the web server and evolved with time. And we thought that promoting glycomics on the XPC server, which was actually used by a lot of other people, especially in proteomics, was a good idea to start. So here we really introduced glycomics in the list of other omics so that it would be visible. And so we use the XPC for those many years, has been collecting resources with two sections, database and tools. And so we develop our own. So when we have a logo, a SIB logo, it's our own. And we also promote even Glycopedias here, others, because we think that we should certainly promote those resources that are useful. So we're working on a new look. This is something I will talk about a little bit more later. And what I want to start with is this depiction of the cell surface. Because mainly we're not going to talk about all the polysaccharides and oligosaccharides and all the sugars that Serge talked about earlier. We are going to focus mainly on glycoprotein at this stage. And I would like to use this picture to summarize the situation of the glycoprotein, the glycans and the lectins, and the glycosaminoglycans that are there as well. So this will be my guide. And we are really trying to collect the information in context. So ideally, if you have a protein, you would like to be able to answer the question, which are the glycosylated sites in that protein? In which conditions are they glycosylated? What are the glycan structures? So what is the repertoire of glycan structures sitting on a particular site? And how much of each, depending on which conditions? So can we have for each a distribution of the glycans that are actually observed and detected on a site, and in which proportion? So this is what we would like to have for a protein, other proteins in the same tissue, so that we can characterize a tissue. And actually, this is our holy grail, and we are very far from it. So if I talk about the glycans, now Kyoko has put together this repository, which is called Glycan, where all the structures that have been reported in the literature and in the databases, they all centralized in Glycan and given an accession number, because it wouldn't occur, I think, to any of you to talk about a gene or a protein without providing with it an accession number to be absolutely sure of which molecular entity you're talking about. Unfortunately, with sugars, we don't have that habit yet. We are enforcing that habit as much as we can, and Glycan is here to serve that purpose. So we are also collecting the information on the glycoproteins in relation to the glycans. And this is the effort that will be shown with GlyConnect and Glygen. So we are also trying, which is a big effort, as you know, there's a lot of repositories now to collect gene expression data, to collect proteomics data, mass spectrometry data that help understanding the identification of protein. But we don't have that very much in the glycomics area. So again, as a collaboration between Glycosmos and our group, we are trying a database which is called UniCard-DB, that was started by Nicholas Carlson, who is in Gothenburg, and trying to have the MSMS annotated spectra to characterize each and every structure of glycans. So we're trying as much as we can to put this data, to get this data annotated in UniCard-DB, and Kyoko has created the GlycoPost repository so that people can submit data instead of having an Excel spreadsheet lost in supplementary material associated with a paper. So we are also helped with the Mirage Initiative, which is supported by the Bellstein Institute, and Mirage standing for minimum information required for a glycomics experiment, which of course for the bioinformaticians who are listening should remind them of all the minimum information standards and guidelines that have been published since the turn of the past century. So we are also trying in that direction to get the information stored in a way that is reusable and understandable by different types of software. One thing we are really weak at the moment is the quantification part. So having different profiles of glycoprotein with the different measurements of how abundant is one sugar as opposed to another, etc. We have the example of the immunoglobulin G, because there's only one glycosylation site. And so a lot of work has been done in that area. And this is the most popular example. So you have liquid chromatography profile where you have glycan peaks And so talk about standards, this is not very standard notation in having glycan peaks numbered in HPLC or UPLC profile. But for IgG, so you have lots of publications showing these sort of profiles that are actually each glycan peak has a number of potential structures behind it. And so now with this, in this simple case, there's a lot of work that has been done, and you have glyco profiles of immunoglobulin, especially IgG. And what is interesting is that what people are trying to monitor is some modification of the glycans. So what is the state of fucosylation? What is the state of galactosylation? What is the state of inserting this glucnac here, which is called bisection? What is the state of silylation? So these terms you're going to see more and more often, I believe, in publication talking about glycoproteomics in particular, where you are actually trying to capture the trends in how is fucosylation evolving, how is galactosylation evolving, etc., etc. And what is interesting is in that paper, which was the study that was led by Manfred Wöhrer and Gordon Lauk, they have actually seen that the ratio of these two, the expression of these two glycans on IgG is age dependent. So whether this is an absolute truth, we are not sure, but there's a trend. There's definitely a trend, and there are things to be captured by just measuring the amounts of glycans in proteins. Of course, it's impossible not to talk about 3D, and a lot of the work that is done. So this is the modeling from the Glycam group, Rob Woods, who is in Georgia in the US. And this is an example of what you find, what Serge does in with Glyco3D. And so these are entries of their respective database, I mean, what you can construct, and trying to here match the SNFG notation. And so techniques of molecular dynamics, of molecular modeling are used for glycans as well. And the idea is that it has an impact biologically, and I cannot escape at this time of the pandemic around the world, not to mention the glycan shield and the fact that all the surface viral protein are highly, I mean, not all the viruses, but the viruses here are heavily decorated by sugars. And the modeling has actually shown that the protein in HIV, you can see the sort of completely different look of the surface protein because it's glycosylated. And needless to say, during the pandemic already, the COVID-19 glycan shield was solved by the among others, the tip of Max Crispin. And this is the model of, so it's actually active research, and this is not really glycoinformatic per se, but it's certainly related, and we need this information. Then we move to binding. And to binding, we need to know about these lectins, so these carbohydrate binding proteins. And we will have tomorrow a day dedicated to this part, talking about the unilectin database that is developed by François Bonnardel, DB, which is focused on pathogen, on human pathogen, and the interaction of human glycans with human pathogens. And we'll have also a presentation on MatrixDB, which actually collects information on interaction of ECM, extracellular matrix proteins, and all the proteoglycans that are described in interaction with glycosaminoglycans. So as you can see, the landscape is populating with databases that talk to each other, that are dedicated to one particular type of molecule, because there's already so much to say, but the idea is really to connect them, to speak the same language, have the same encoding, have the same format, have the similar representation, so that we can communicate data and then the users benefit from this. We also develop tools. So I will say a few things on the tools that we have, and I assume that Kyoko with Glycosmos and Raja and Mike with Glycan will say a few things on the tools that they develop as well. So in the end, I'd like to insist on the fact that a lot of the progress in bioinformatics has stemmed from challenges, community challenges, like the CASP challenge in the prediction of three-dimensional protein structure from the sequence. There was a number of editions, there were a number of editions of this challenge with some data offered to the community, to all the developers, to benchmark and to improve their software. So we need to do the same in glycomics, and reproducibility is already an issue because detecting sugar is so difficult. There's so many techniques. So very recently, there was an interlaboratory study on the glycosylation of monoclonal antibodies. So you can see that there were a lot of participants. And as you can see, in some cases, so we're talking composition, so I'm giving you some example of the structures that are attached to composition. You would have noticed actually that when I mentioned some sugar structure from 2D to 3D, I had glycan accession numbers for each of the structures that I mentioned. So I have the reflex that I'm trying to transfer to you. Here, I didn't put it because it was just an example, but I should have. And you can see here that all of the labs and all of the techniques, the different techniques, whether it's liquid chromatography, capillary electrophoresis, they are sort of finding the same composition on the monoclonal antibody. So there's a strong correlation for that as well. But with less abundant composition, then you have scattered results. And this is where there's a lot of work still remaining to be done at the experimental level to enhance data reproducibility, so that in the end, when it ends up in databases like ours, we are somehow providing reliable data. Another, of course, challenge, which is software related, has been launched last year by the during the HUPO by the Human Glycoproteins Initiative, and so some human serum sample were actually analyzed by the organizers of the competition. Then they generated the data, and it was analyzed by 9 teams who develop software and 13 teams who use software. And we are very keen to get the result, which we had just a glimpse of during the HUPO conference last year. And again, reproducibility is an issue, as we could see from the preliminary results. So we will have to improve software with these sort of challenges. So I'm done with this presentation. I have to thank most particularly Julien Marietto, Thibault Robin, and François Bonardel in my group for developing the hopefully nice resources that we provide. I also cannot say that we cannot work in isolation, and so we have a group of very close collaborators who have different specialties. So of course, Serge and Anne in Grenoble, Nicholas Carlson in Sweden, Sylvie Ricard-Blum, who is in Lyon and who will talk tomorrow, and last but not least, Nikki Packer and Daniel Kolarich in Australia, with whom we started quite a while ago discussing all these glycoinformatics issues. I appreciate very much the enthusiasm of Kyoko, who promoted the GlySpace Alliance and really created the momentum for that. And that was really, and she will talk about what she's doing. And I also thank Elizabeth Gasteiger from the Swisspot group, because she is really our communicating channel, opening the communication channel between UniProt and our resources. And I also thank the community for using our resources. These are our sponsors, and I thank you. You can follow us in the news we give on Twitter.